Ok, buongiorno a tutti e tutte, benvenuti e benvenute. Oggi è in inglese, quindi ora inizio a parlare in inglese. Today we have the pleasure to have Andrea Fiore from the Netherlands, Eindhoven University, Technical University of Eindhoven, and he's an expert of optics in many problems. Today he will speak about a new sensors, but this is something he will explain to you. As usual, I go explaining some details. So you will find in the chat the link for the form you have to fill in order to have a certification of your presence. So please go there and fill the form. In the form, you will find 3 questions that Andrea prepared for you. They are in Italian, so this is simply for you. And there are 3 answers, so only one is correct as usual. So you have to answer this question at the end of the webinar, of course, because listening to the webinar, you will learn the answer. And the next webinar will be again on something related to ophthalmology and optometries. So it's an optometrist from the University of Milan that will explain us what an optometrist has to know to help the surgery during a kind of operation to the eye and things like this. So it's a subject that I think for our student from the university in optics and optometry is quite important. Coming to today, so what Andrea will explain us is a new chip that they build up in order to have an optical sensor of materials. And as usual, I asked to Andrea to prepare the first part, which is quite didactic and as simple as possible, just for you that are not so expert in the field. And after 20-25 minutes, Andrea will stop and I will look for any question from your side. So you can write the question in Italian if you want. Andrea is an Italian friend, so he understands very well Italian, but we expect to have also some guests from Europe, so we have prepared the webinar in English, but you can write down the question in Italian. There we reply in English, but if you are more confident, you can write in Italian. And then he will continue, and at the end of the webinar, we will have an extra time for discussion, about half an hour, and we will wait for your questions, and then we will have also 3 friends and colleagues from university. Anna Vinatieri is professor in many fields of optics and semiconductor optics, which is the core business of the sensor. And Marc Abarchi is from the University of Marseille, and he's also within a spin-off that is related to nanotechnology and things that are very strictly related to the subject of the today's webinar. So with this, Andrea, I have finished my talk and presentation. So it's your time, please go ahead.
Yes, good afternoon, everybody. I think you're seeing my screen now.
Yes,
correct?
yes, everything is fine. Yes.
Yeah, well, thanks a lot, Massimo, for the introduction and kind invitation. I'm honored to take part in this long-standing webinar series. So today I'll be talking about near-infrared spectral sensing, and I will have a subtitle, The Invisible Colors of Light. This is basically work that was done mostly in my group at the Eindhoven University of Technology in the Netherlands, but also in close collaboration with our spin-off, Spectra. We tell you more about this in the course of the lecture. Also, for transparency, I put up a statement of potential conflict of interest. I just mentioned that a few of the authors, including myself, are also shareholders of the company. Okay. Well, I wanted to start maybe with a little marketing for our university. So we are in Eindhoven, that's a city in the south of the Netherlands. Mostly know about for PSV, the football team, but we also have a great actually university, it's an engineer university equivalent to Politecnico in Italian system. It's a mid-sized university, about 14,000 students. We have English as an official language, so that means we're also very open to international students, and we are particularly good in applied research and industrial collaboration. We are systematically ranked first or second worldwide in terms of industrial collaborations. And that has to do a lot with our location in industry. So I guess you may know Eindhoven just from PSV, but actually PSV exists because Philips was born in Eindhoven. And that's a trigger, well, the formation of a very important high-tech hotspot. So we are one of the biggest hotspots in Europe for technology. And it's not only Philips, but also ASML, Thermo Fisher, and many other companies. And that means that there is also very, very good job market for our students, master students or PhD. And because this industry is also looking for people who have a specialization in optics and photonics, we are also ramping up our efforts to educate more people in this field, particularly in the master program. From next year, we have a special program which we call Master Plus Optics and Photonics. And it's a master program, so it's a standard master program with a plus, so a range of activities that bring students in closer contact with our industrial ecosystem. If you have any interest now or in the future, feel free to look at our website. Okay, now for this talk, basically I have three parts. First, I will give a very broad introduction to spectroscopy and spectral sensing. So what is it and why is it useful? Then I will present what we developed. So the chips that Massimo was referring to, this is a basically very simple hardware for spectral sensing that essentially in a few words will hopefully replace a spectrometer for many applications. And then in the third part, I will go into some details of how can we use it? So what can we measure? So let me start with spectral sensing. Effectively, I would define it as using color information to measure or characterize materials. And this is something that we do daily, actually every second with our eyes. We look at the color and we use this to take decisions, to buy, select things in the supermarket, to determine whether fruit is ripe or not, something sweet or not. I think you see a nice example in this image. Now, you know that this is done in our eyes by specific photoreceptors on our retina, photoreceptor cells. There are different kind of cells that contribute to vision, and cone cells are responsible for color vision. And there are 6 million of these, and that allows they allow us to, first of all, to of course see an image. So to image the scene, but also to get the color information. And that is done by actually 3 types of cells, say red, green, and blue. And we can see in this plot a little bit more how they work. So basically, one of this type of cells is more sensitive to light in the blue part of the spectrum. So here you see the response of the cells as a function of wavelength. And of course, this is the visible range from about 400 nanometer to about 700 nanometer. So we have the blue, we have the green cone cells, which measure more in the green, and then of course the red one more in the red. Of course, it's not, they have a lot of overlap as well, but they cover slightly different ranges. Now, of course, the information from these cells is communicated to the brain and is combined together as what we call color. And what is interesting is that we actually build throughout our life experience by just, by experimenting, by seeing, by tasting a red strawberry, we know that it's sweet, and tasting a green one, we know that it's not sweet. And so these are kind of learning. And please keep this in mind because this will come back in my talk. Now it's also interesting to understand why we get colors from different colors from different materials, and this is mostly coming out of diffuse reflection. So maybe good to remind ourselves that reflection can be specular, and then you just basically probe the surface, but it can also be diffuse. And the main difference is specular reflection, of course, occurs at the same, well, reflection angle. The reflected angle is the same as the angle of incidence, whereas diffuse it's basically covering all angles. And that is actually the reason why it's so important, because we all, of course, we need that. Otherwise, we would only see light that is, well, reflected from a very specific angle, let's say. Now, what produces diffuse refraction? Well, light that is incident on a material will be partly transmitted, and we propagate through this material. But this material is typically not perfectly homogeneous, so there are some inhomogeneities, and those will scatter light. So, and light can be scattered actually multiple times. You see this theoretical rays. And by this process, it can be basically reflected back, so it can be sent back to air at different angles. Now, this is actually very crucial, because this light has traveled through the material, and these paths are quite complex, because of course the scattering is random. It can occur in many different ways. So there are basically many different paths with different lengths, and we like to understand this, approximate this as saying that there is an effective medium average length of propagation in material, but of course it's more complex than that. Now, what is important is that, of course, the length of propagation is important, because in this propagation there will be absorption, and there will be this absorption depends on wavelength, because the material absorbs some wavelength more than the other. I will come back to this in the next slide. And of course, the product of the absorption coefficient and the effective length, this is essentially what sets the total transmission. And that is also what actually gives the spectrum of reflection, because if light is absorbed, well, we don't see this in the reflected in the spectrum diffuse reflection. Okay, so basically the key message here is that diffuse reflection, even though it's reflection, actually gives you information on the transmission, on the absorption spectrum, transmission and absorption. And that is something that you see in all cases, basically. So if you measure the spectral reflected light, you will see modulation, and you will see spectral features like dips, or regions where there's very little reflected light, and regions where there's more reflected light. And that all gives information on this absorption coefficient, and therefore on the property of the material. So color is directly related to the property of the material. And of course, we knew that, and this is also a way we actually use to even characterize the material. So see what kind of material it is. We can normally say quite easily is that something wood or plastic or so on. Now, microscopically, we can look at the origin of this absorption, and that has to do mostly with vibrations. So vibration of molecular bonds. So if you take something as simple as water, well, the molecules that water molecule is made of atoms, and there are bonds between these atoms, and this will vibrate. And there are many different type of vibrations that you can define in such a molecule. They are more and more complex as the molecule gets bigger. But the fundamental physics is basically the physics of spring. So if you have a simple spring, well, typically, so it will have a natural frequency that is related to the spring constant of the mass. And what is important is that if you drive this spring, imagine just periodically pushing against it at a certain frequency, well, you can drive it effectively only if you match its natural frequency of vibration. And it's at that frequency that you actually transfer power to the string, or to transfer the maximum power. And this is actually exactly what's going on when light wave impinges on a material. The electromagnetic field exerts a force on the charges, particular electric force, and that will be most effective in producing, in transferring power, where the frequency of the wave matches the natural frequency of these vibrations, or a multiple of that. So that is why we see some frequencies or some wavelengths that are more absorbed than the other, because they match those natural frequencies. Now we can have an overview of what are the relevant, well, wavelengths or frequencies. In this case, it's about wavelength for this vibration, vibration absorption by looking at this plot. And of course, here we see particularly the near infrared range. So remember, you have the visible range is between 400 and 700 nanometer approximately. The near infrared, there may be several definitions, but here I will use the definition which is standard near infrared spectroscopy, and that is between 700 and about 2500 nanometer. And well, actually, if you look at the natural oscillation frequency of these bonds, they are actually beyond even this. So it's at longer wavelength. That is what we call mid-infrared. And so this is called the fundamental band. So there you get the maximum, the strongest signature, the stronger absorption. So in principle, it's the best one. However, it's very difficult to measure in this wavelength region. It's not impossible, but the equipment is very expensive. That has to do with the fact that it's difficult to make these detectors, basically. Then there are multiples of these frequencies, so the harmonics, and these are called typical overtones, and the first overtones are in these infrared regions. And you see that, for example, so each type of bond and therefore each type of molecule has a signature in several bands, in several, say, spectral bands in this region. So basically that tells you that if you're able to measure in this region, you should be able to retrieve what kind of molecule it is. Although it's still complex, you see, because there is overlap and each molecule has many actually of the signatures, so it can get pretty complex, but still there is a lot of information here. Of course, there's also information at shorter wavelengths, so more towards the visible, but then you are talking about higher and higher harmonics. And then it's a weaker signature. So then if we look at the technology, well, we have, of course, human eye that is sensitive here. So of course we can get information, but it's not often difficult to get a quantitative information. Then we can go with silicon technology. So the same technology that is used in our computers in CMOS. That is, of course, a extremely well-developed technology. It's the technology of CMOS cameras as well. So it's very convenient. However, it covers up to about 1100 nanometer. And the reason is the band gap of silicon that is at that wavelength. So at longer wavelength, basically you don't absorb anymore in silicon. And then you have to use other material, and particularly 3-5 semiconductors, and this is particularly the material technology we are working on in Eindhoven. Okay, now, of course, the question is, well, we have our eyes. Is that enough? Can we do better? And yes, we can. You can imagine we can do better because we measure only using 3 wavelength bands and only in the visible. And we have seen that most of the information is actually at longer wavelengths. So the first thing is, okay, is there any system in nature which is better than human eye? And the answer is actually yes. So there are some animals that have better color vision, or at least more, that can sense more wavelength bands. And actually the record holder is the so-called mantis shrimp. It's a shrimp living in the sea, and it can measure up to 12 wavelength bands. So it has a much better color resolution than we have, and actually it seems it really needs that for its own survival. Well, it's an interesting animal. It's also a very, very aggressive animal. So you can also get bites from the shrimp. But from the point of view of color vision, it's really the dominant one, or the best one. Now, of course, we have been also as humans working on making instruments that can do this better. And these instruments, as you know, they are called spectrometers. So basically, the general idea is very simple. You come with white light or whatever light you want to measure the spectrum of, and then you disperse it using, for example, diffraction grating, but you can also use it prism or other instruments, and that will basically separate the different wavelengths spatially. And then what you do is, well, you put a slit, so a screen with a small opening, and then you will select only one wavelength, and then here you have a detector, for example. or else you can put a camera here and then measure all the wavelength at once. So this is a standard way of doing spectrometry. It was developed actually in the 19th century, so it's about 200 years old technology, and it's interesting enough. The same, of course, the spectrometers have evolved, but essentially the same physical principle and the same approach to spectral measurement is still used today. And it's used in, of course, it's used in a research lab, but also used a lot in industry, in this kind of industrial spectrometers. Just because of what I said, with my measuring the spectrum of light, you can actually say something about the properties of the material. I will come back to this later. So how can we use the spectrum to measure, to effectively say predict some property in practice, let's say. But for the moment, I think you see that there is a relation between the spectrum and the property of the material. Now, of course, this is again a very successful technology. But now in the last last years have come a little bit into pressure, let's say, and the reason is there is an increasing need in industry, but also all the way down to the consumer application and healthcare, to actually have a measurement of the material not at the end of a process, but in line during the process when you are actually producing. And a simple case is, well, you have when you're in the field and you want to harvest, you would actually want to know at that point where it is the good time to do that and where maybe. And similar thing for healthcare. In many cases, you would like to have a prediction on, to know whether a wound is dangerous or not, maybe know more about what's going on in our body. And even in consumer application, there are cases where you would like to measure what kind of tissue I'm putting in a washing machine, or what kind of material I'm using for preparing, in my kitchen and so on. And so this is coming up more and more. But of course, in this case, you need a spectral measurement, but you need it in a very small footprint, something that you can integrate in a bigger system, and it has to be very low cost. And that is extremely difficult using standard spectrometer, just because you have to put together this grating and the slate and the detector and so on. This is assembled, and it's difficult to scale this cost and size down. So for this reason, there's been quite an effort in terms of research, in trying to offer the same functionalities as a spectrometer, but on an integrated chip, so that can be fabricated like our CMOS cameras or computer chips using a scalable semiconductor fabrication technology. Now, again, there is a lot of effort in this area and a lot of very nice results and approaches. I'll show here one. This is not from our group, but effectively one basically category of approaches is we take the incident light, we couple it to a so-called photonic integrated circuit, which is basically a circuit where light is controlled on a chip. And then here, for example, they use a grating, which is integrated in this chip in order to separate the different wavelengths. And then we have an integrated array of detectors, for example. And there are many different variations. And these spectrometers can be extremely performing, for example, in terms of resolution. However, we have to be a bit careful with the way of using that. And particularly the problem is that you need to couple light to a waveguide in all these approaches. And now there is a very simple theorem in optics that tells you that the product of size of an image and its angular size, so the numerical, what's called numerical aperture, angular aperture, cannot be changed by an optical system. And now the problem is this waveguide is a few micron in size, but the area from which you are typically collecting light from an object, if you do diffuse reflectance, you are in free space, you are a certain distance and so on. So we're talking about typical centimeters, and it's completely impossible to focus on the light efficiently into a waveguide like this. So that is a bit of a warning that many ideas and concepts, they are only applicable if your light is coming already from something like a single mode fiber. And that is a very small minority of the practical application cases. So for that reason, although we started working with this kind of approaches, at some point we switched to something different. And what we did was to start to just integrate on a chip an array of detectors where each detector would measure a different part of the spectrum. And this is actually, as you see, very similar to the idea of the cone cells, right? The separate cone cells measure bands. And to some extent similar of the idea of just RGB camera, but then expanded to 16 pixels with a much higher resolution and in linear infrared. And of course, the nice feature about this approach is that, first of all, you collect quite some light, right? Because this can be a few millimeter in size. So you don't need to focus light on a couple of microns, but it's still a very small device. So its total chip is a couple of millimeter in size, and it can be produced at mass in a semiconductor fabrication process. So you can produce basically hundreds of thousands of these on a wafer, which means that it's both small and potentially low cost. And yeah, this means that if we're able to use this kind of system for spectroscopy, then spectroscopy comes basically at your fingertip. Okay, now let's see how we can make those. So essentially, they are photodetectors that are relatively standard in the type of diodes. So this is essentially an InGaAs photodiode. What we really do on top of that is integrating a filter. And we do this by putting a bottom mirror and a top mirror. And these two mirrors together, they form a Fabry-Perot cavity, and that produces an optical resonance. And this is what we use because light that is incident from the top can only get inside the structure if it matches basically the resonant wavelength of this cavity. Now this is and the other thing we do, is we can tune the wavelength of this cavity by changing the thickness of this one of these layers, and then basically we have a different optical length for each pixel, and that makes the pixel respond to some wavelength, for example. Now this is in principle, the principle is very simple. There was one big technological problem, and that is that the fact that this bottom mirror actually cannot be grown epitaxially, so it cannot be grown together with this part. And the reason is that the material is quite different. And for that reason, we need to develop a specific fabrication technology where basically we start with this indium phosphide InGaAs. This is a 3,5 material that we can use to make this photodetector basically. And then we deposit a silver mirror on it. Then we flip it. We bond it. So we basically just press, we can glue together with a carrier substrate, and that is typically silicon. Our glue is a polymer, it's PCB, and after this, well, you have your 3,5 wafer on top of the carrier wafer, and then you can etch away the original substrate, and then you can start to process it from this side. And so it's still, you can process it, but the thing is we now have the bottom mirror, and we can simply add a top mirror on top, and then we have the entire structure. Before we break, just a quick overview of the kind of response that we get. So this is an example of the responsivity. So that is the ratio of current to power. So this is how strongly basically our detector responds to as function of wavelength. And you see that so each curve, each color corresponds to one specific pixel of the array. And you see that we have a clear peak in the spectrum, and that is because of the resonance that I mentioned. So only the wavelength, only this wavelength can be coupled into that cavity and therefore be absorbed. Each of these peaks has a width of typically 50 to 60 nanometers, so it's relatively narrow, but not as narrow as you would have in a spectrometer. And another thing you see is that each pixel actually has multiple peaks. And that is what you can expect from this kind of structure because you have multiple resonances, actually. There's also an envelope, but this is due to the standing wave pattern that we create in the structure. So between the two mirrors, there is a standing wave, and then there's a minima, maxima, and they overlap more or less with the absorber region. And so that's the reason why some wavelengths are a bit less absorbed than others. Okay, one thing I want to stress is that this is not a spectrometer, right? Because we are not measuring wavelength by wavelength. We are measuring only in certain bands, and even one pixel is measuring actually multiple bands. Okay, now I would like to stop here for a short break and maybe ask if there are questions until this point.
Thanks, Andrea. Thanks. I ask the students from home to make questions. I can't see from now, so I used to do some questions. So the first is why 16 sensors? So why not 12 like Mantis or 20 or whatever?
Yeah, it's a good question. I mean, there is no there is no very fundamental reason. Obviously, at some point, it's a kind of engineering choice that you make. But when you look at this spectrum, I think that gives you an idea of why. So we want to cover the maximum possible range that we can cover with this material, and that is limited by the band gap of the absorber, which is around 1700 nanometer, and well, at shorter wavelength, there's other materials that start to absorb a lot, and you see that the response goes down. So this is the other limit is about 900 nanometer. So we have a quite wide range. This is very useful because it's very useful for applications to cover a big range. And now, if we had a much narrower line width, then we would need indeed many more pixels. Well, we would have a better spectral resolution, so that could be an advantage. However, then it starts to get a large array. Now, large array means that also you need a lot of electronics, complex electronics, to read it out. So 16, it's well, beyond 16, things get significantly more difficult. You could do less, of course, but then you lose in terms of well, you lose more, you lose spectral resolution, so then you have to compromise a bit more. So it's I don't actually the answer is the real answer is depends on the application. So if we for some application 3 or 4 would be enough. For others you may need 50, and this is a bit of a trade-off.
Okay, and then just a comment, maybe you can add something. So any molecules has a kind of barcode, like when you go to the supermarket, any product has a barcode, and then you can recognize the product with a barcode. So you are doing the same, more or less.
Yeah, I would love that it would be as accurate as a barcode. Unfortunately, it's not. So the spectra, so I will show some spectra also later, but you will see that the spectra are, well, there are some features, but they are quite broad, and so it's not as, the information is not as detailed as in a barcode, but that's the idea.
Listen, Andrea, there is a question. I don't think is a student, but anyway, Thomas is asking you, Thomas Kraus, you can see, is asking you, what is the angular dependence of your resonator detector, given that you mentioned conservation of ATNU?
Yeah, well, first of all, nice to have you here, Thomas. Yeah, angular dependence. Yeah, we have looked at this. I don't have a slide, but it's in our published paper. So this paper, there's angular dependence. This is a critical point indeed. And this is actually the main reason why we chose for a very simple Fabry-Pérot structure, because it is among the things that you can do easily, is the one that has a relatively low angular dependence. It just goes as a cosine. So that is, for that reason, a quite well, good choice. In absolute terms, I think for an NA up to 0.2 or so, there is for this, of course, for this line width, there is a negligible wavelength shift with the angle. Of course, if you go beyond, you will see it. It's not necessarily a problem as long as you have the same optical system. So if you are measuring the object, and the object is always, for example, in contact with well, your module box or whatever, so that means that the angular distribution is also more or less constant. That is fine. Of course, if you're pushing the limits, you will have problems at some point. But the fab repair is actually doing very well with respect to many other, well, of the, I'm not saying all, so I know that you have been working on also angular, well, flat, and you can engineer dispersion relation, but if you, the most simple photonic crystal structure, they will have a lot more actually angular dependence.
Okay, okay. I think you can start again in the presentation and the webinar. So please go
All right.
ahead.
Okay, so part two is more about how, what can we do with this kind of device? And that was an important question because our device is not a spectrometer. So we cannot claim that we have a spectrometer. We have to claim that we can do something with it, something else. And that was a very good exercise also for me to go into understanding how you use this data. So the idea of a general experiment is the following. Of course, you have your light source, you have an object that you're measuring, and effectively you then will somehow focus, bring light, which is reflected onto your array. Now, the object has a certain spectrum, and that is the spectral information that you want to use. What you do is, well, each pixel of the array has a different spectral response. In the end, you collect, in our case, 16 photocurrents, and of course, each photocurrent depends on the spectral overlap between your incident spectrum and the response of that particular pixel. Okay, now from here, you can go two ways. So one way is you really want to have the spectrum, and then you can apply some numerical methods in order to try to estimate your spectrum based on this information. And of course, this is a kind of ill-posed problem in the sense that, well, you have 16 measurements and you need to retrieve maybe 1,000, I don't know how many wavelength points you want, but this is, you have to make some assumptions. You can still do that under some assumptions, and you can try to estimate, reconstruct the spectrum basically. And then from here, of course, it's an easy ride in the sense that there's a large field of statistics, it's called chemometrics, where basically there are ways, the methods in order to establish a prediction model, which using, say, a series of reference measurements. So what you will have to do is measure, well, in this case, hundreds or so, in any case, several tens up to hundreds of samples, say tomatoes. You measure the spectrum and you independently measure the parameter that you're interested in, let's say sugar content. And then you have methods that basically will build a prediction model based on this association relation between the spectrum and the parameter. And eventually you can have a predictive model. So you come with a, well, unknown object, you measure the spectrum, and then the model will estimate the, for example, sugar level. So this is well known. However, when actually I looked at how this work, it was very interesting to understand that the first thing that these models do is to try to simplify and reduce the number of data. And because the reason is the spectra are pretty broad in general, you see that. And that means that many wavelengths that are close to each other do not have independent information. And that is a big problem when you're trying to create a predictive model, because you have the risk of creating a model which is actually just trying to fit the noise, basically. So, and effectively what they do is they basically project this into a basis of typically 10 or so called principal components, or it's principal components, and then use these to build the model. Now, when I realized this, we understood, okay, this is not the way to go, because here you are expanding your data and then you are compressing it again. And in the end, that is what you have. So for our hardware, this is the only thing we have. So it is our best shot is try to use it directly to estimate the parameter. So we use basically the same methods and also the same software packages in order to build this predictive model that uses directly the 16 photocurrents as if they were, well, the measurement of a spectrometer together with the reference data in order to build a predictive model. And now the reason why it is logical that this can work, at least in principle, is that this is of course what we do. with our eyes, with our brain, we learn, right? And we never reconstruct spectrum in our brain. We just learn what color is good and what is not good. So that was also a motivation to try that. Of course, there is a complication when you want to do this, that you have basically to do the entire thing, because you have your spectral chip. Okay, that's the beginning. Where that is typically where we would end as a group, right, as a research group. But that's not enough. So you want to show that it works. Then, okay, you have to build an entire sensing system in order to make measurements. And so we started this originally in my group. Then it was pushed forward by the spin-off, and they built this nice modular with basically light source, electronic readout, connectivity, everything. And now it's this module is actually the previous version. Now they have a smaller one. And with this, you can start to take data. And then, of course, you need also the models, the methods to build the model and then make prediction. So that was actually a quite interesting experience to try to make all this. But this is now, of course, fully, spun off in our group, by the way. The spin-off is called Multispectra, and you may recognize where the inspiration for the name came from. Okay, now in practice, what does it mean? Well, we and the company have been experimenting with many different application cases. I will showcase a few. One is, it's very simple. It's a very simple problem, but it's also a very, very useful problem. It's measuring water content in materials, and particularly biological organic materials. And this is an example. We took a simple one, rice, and we try to build a model for estimating water content by basically soaking the rice in water and then drying it, and then you can measure at different weights, so you can quite easily measure the amount of water. And basically, you can do it with a spectrometer. So we almost always do this. We take a commercial reference spectrometer, and we measure the spectra. From the spectra, you see clearly some few spectral features which are related to water absorption. And at the same time, in the same campaign, we do it with our spectral sensor. And here, of course, again, we just have 16 photocurrents. Then we can make a prediction model using the spectrometer. And this was a result for this problem. And you see this typically this relation between the predicted value and the calibrated value. And you do this with using calibration data first. So of course, these lines are these points are typically quite well aligned. And then so you also do it with test data, so unknown data, basically completely new samples. And that works also quite well. Of course, there is some error, but that's also normal for this kind of problem. But it's a good prediction model. We do this with our spectral sensor, and we find that we also have very good correlation between the prediction and the calibration. Error is slightly higher, so this is error prediction. In this case, is 1.4%, with the spectrometer 1.1%. Of course, this will depend a lot on the problem, a lot on experimental procedure, a lot of what you do and what you measure. But overall, this is a bit the message that we have, that we can typically get results for this kind of probe, sensing probe from solids and liquids. We can get comparable prediction, a bit worse, but in the same range as with a complete spectrometer. Classification of plastics is a very, very nice problem because it's simple and it's also interesting. So of course we know that, we see the color of plastics, but the color of plastic has nothing to do with the type of plastics, because of course, they put a paint in order to look nice or whatever. So in the visible, you really cannot do it from the color, but the near infrared, there you really sense the relevant chemical information. And that works extremely well. So this is well known to be possible with spectroscopy. For example, we have a classification here between these 6 different types of plastics. And basically, in this case, it was 100% classification. So very, very easy. And this is also something that we bring this along as a demonstrator in exhibits, or it's very easy to show people and show what this can do. Another relevant problem is actually the measurement of the nutritional properties of milk. So this is done by basically measuring fat in milk, for example. Very important because the value of milk depends on its nutritional property. Also working very well here. So this is also a problem which is known to be simple. with near infrared. When we look at another property, for example, protein content, well, then, yeah, we see some correlation, but it's much more difficult and there's a lot more error. And this is, again, also well known. So again, we are finding that we are basically we're doing, we have similar possibilities and problems as a standard near infrared spectroscopy. Okay, and there are many more. So this is, of course, now it's say it's being commercialized, and there are also many more application cases that are being studied. Okay, now I would like to dedicate a few more minutes to another topic, and that is another use of the spectral sensors, and that is to measure something that has been, well, built in order to transduce some parameter of interest into the spectrum. I'll give you one very famous example that we'll all hate, of course, is a COVID self-test. And you know that a red bar means that some specific biomarkers related to COVID have been detected. And this color change is what is used. So it's a transduction in the sense that the presence of given biomarkers results in a change in color, and then we measure that change of color with our eyes. And this is actually an approach that is very often used in sensing, and particularly in biosensing, but more generally also in fiber sensing, for example, where we design a sensor where what we need to measure will be transduced in a change in the spectrum. Let me show this particularly for biosensor. Now, biosensing here, I'm going to refer to the case where we want to detect very low concentration. So not say the COVID cell test, but we want to quantify the concentration of a given biomarker in, for example, our blood. And one typical way of doing this is we take a surface, we define a functionalization layer with a layer of bioreceptors, and then we try to detect. So this will basically bind selectively the target molecule that we want to detect. Typically, will be a protein that will be basically binding only if, well, only that protein will bind. And then we have an optical sensing system. We try to measure this optically because that's the most sensitive way of doing this. Now, again, this part is what I call transducer, and the goal is to have a structure that has an optical property such that, well, it produces a given spectrum, and in this case, very simple, is a peak, right, in the spectrum, and this spectrum will shift if there is this additional layer of proteins being bound. And basically, if we can measure this, then we can measure the presence of that biomarker. The second part is the readout. So how can you actually measure this small wavelength shift? And of course, for some cases, for many practical cases, you want to do it in a relatively simple and inexpensive way. So using a spectrometer in this case is not a good idea because it's very expensive, especially particularly if you want to make a kind of point of care diagnostic solution. Of course, there could be different ways of different tricks of doing the job. Well, Thomas was in chat before, is working on some of this. In our group, we tried something different, and our idea was, okay, well, there is spectral resonance, and we want to measure a small change in it. Well, can we do this with a very simple hardware, particularly that basically array of detectors that we can integrate on a single chip. So can we measure this wavelength shift using a very limited set of spectral bands? Let me go in a bit more detail. So the idea of the sensing system is we have a broadband light source. We go to the optical sensor or transducer. We get a reflection spectrum where there is a clear spectral feature, let's say a peak, could be a dip, could be something else, but something that will shift. And then we want to measure this with this multispectral readout chip. In this case, we'll use only 4. And then, well, before Massimo asked the question, why 4? Well, it could be 2, actually. 2 is really needed, actually. It could be more than 4, but it's a good trade-off for several reasons. Then, yeah, and then we will place the response. So I don't go into detail of this, but basically it's the same kind of structure that I showed before, same kind of device, resonant cavity detector. So, and then we would place responses of the 4 pixels around the peak that we want to measure. And this is actually a measure reflection peak from one of these sensors. And then the idea is very simple. Well, of course, the photocurrents that we will measure depend on overlap between our reflected light peak, reflected light spectrum, and the responses. And of course, if this peaks shifts spectrally, well, the currents will change, and particularly the relative ratio of the currents will change. And that is crucial because we need to be insensitive to power fluctuations. That's why we need at least two. A single one would have too much noise. But if we use the relative ratio of these photocurrents, then we can get rid of a simple power fluctuation. Okay, well, the next step is to build, again, a prediction model. So in this case, we don't use any fitting or anything like this. We just take 4 currents. We train a model, so we measure it for different value of, we produce a shift of the peak. We measure it spectrally, say, with an optical spectrum analyzer. Then we correlate this with the 4 photocurrents, and then we build a prediction model that will allows us to predict the wavelength based on 4 unknown currents, and that's basically on a least square principle. And you see that this works extremely well, so that we can have very accurate determination of the wavelength, also for test data. But the most interesting is actually to look at what is the error or the imprecision of our measurement. And so basically, if we keep everything stable, right, we will have some fluctuation. So, for example, even if we measure with the OSA, when we measure the spectrum, we try to get the wavelength, and well, we'll have some fluctuation. That's an error in the measurement. Well, the interesting thing is that that fluctuation is actually lower when we use our simple multispectral chip than when we use an optical spectrum analyzer, so a spectrometer, which, by the way, is an instrument that costs about 30,000 euros. And the reason is actually, so you can prove that there is a very simple fundamental reason for that. So you can make a very simple theory that shows you that you will have a better resolution, a better precision, just because you have a much higher signal to noise ratio. And this comes with the fact that we have a large area of we only have 4 pixels, and they each get quite some light. So this is actually predicted by theory. Quantitatively, of course, how much noise we get depends on the integration time. So here we quantify with so-called Allen deviation. And we see that if we use an integration time of a few seconds, we can get, in this case, below 10 picometer in wavelength precision. This was with a photonic crystal transducer. Using other kind of transducers, we got below 1 picometer. And that is, again, much lower than the best you can do with an optical spectrum analyzer and a fitting, for example. So this looks good because this is the range that you need of precision that you need for biosensing. So of course, the next step would actually to show that this can be used for biosensing. We used a very simple assay, well-known assay. This is basically using immunoglobulin G. In this case, we use it as a receptor, and we use its anti-IgG, so it's antigen, as an analyte to detect. As a sensor, in this case, we used a 2-dimensional photonic crystal. I'm not going to go into detail how this works. What I want to point out is that this produces a reflection spectrum with a clear peak, but also other peaks that we really didn't want, actually, in this case. And also there is a background, and this is caused by our sensing system, which is not optimized. So you see, this is not the kind of spectrum actually you would ideally want, but I will show you that even with this, it actually works. And then, of course, we place our spectral responses for the multispectral array around it. Now, how does the experiment work? Well, we basically have a first part, which is calibration. So we basically have this inside a fluidic cell. We flow several liquids with different refractive indices, and we see that the wavelength shifts due to the refractive index of the liquids. We measure that with the optical spectrum analyzer and with our multispectral sensor, and we use this data in order to build a model for the rest of the experiment. Then in the rest of experiment, we first flow the functionalization layer so that we basically bind these capture molecules on the surface, and we see that this produces the change of surface, so we see that. And then after this, we actually have the real bioassay, so we flow, in this case, the anti-IgG, and we see that this actually binds on the surface, produces a shift, which is of course a fraction of a nanometer in this case. And what is interesting now is not so much the absolute performance in terms of biosensing. I mean, this was, if I recall correctly, about 100 nanomolar. So it's not yet at the level where you can detect very small concentrations. However, I'm more interested in seeing that our estimation, which is done with the multispectral readout, follows very well the wavelength that is measured by the OSA throughout this experiment. And if you look at the also precision, it will, well, I showed you before. So basically, it's actually even in general more precise or comparable depending on specific case. And that basically shows you that you can have a precision readout that is adequate for the readout of these biosensors. Now, in conclusion, very short message take home. These spectral sensor, integrated spectral sensor, they are not spectrometers, but they can actually replace spectrometers for many practical cases, in particular near-infrared spectroscopy, characterization of materials, but also for high-resolution optical sensing or high-precision optical sensing. So also for measuring extremely small changes in a sensor that are produced by molecules bonding, small temperature change, we can be extremely precise, despite the fact that the line width of these sensors are relatively large and much larger than the spectrometer. With this, I would like to thank you for your attention. I'm looking forward to more questions.
Okay, so thanks, Andrea. Thanks for this very nice talk. And I'm looking for questions in the chat, in YouTube chat. So far, there are no questions. So again, please ask questions, whatever question you want to ask. I mean, there are no stupid questions, I used to say. Anyway, let's start with our expert. Anna is the first.
Thank you, Andrea, for your very nice and interesting talk. And I have a question. You can reach a very high sensitivity in the spectrum, so you can measure the wavelength in a very precise way. But my question is, which is the detectivity limit in terms of intensity of your signal?
Yeah. Thanks for the question. We have measured this and published this. If I recall correctly, what we have at the moment is in a few tens of picowatts as noise equivalent power per square hertz. This is at the moment not really optimized, so we know that we have more noise in our photodetector than we should have with optimized photodiode for these wavelengths. And this is a matter, I think it has to do with the process. And on top of that, of course, there is also noise from the readout. So there, of course, is a trade-off between what you can spend, let's say, on the amplifier and the sensitivity or the noise equivalent power. So I think these are, in general, they're very good detectors, but they are still far from any fundamental limit. So you could push this down by probably at least one order of magnitude and maybe two. And that is, in many cases, actually, okay, one thing to realize is that it depends what you're measuring. So for the experiment that I show at the end, so they're using them for a readout, and also for many spectral sensing cases, you're not limited by that. So what you want, because you can put quite some power in your lamp. And what we're more interested in is a really signal to noise ratio. So, and in many cases, that's fine. And then you will, if you had much lower noise, you maybe run into problem of just of dynamic range. So you have to also convert to say digitally and so on. So that is, in many cases, that's not the NEP is not the limiting factor, but there will be a few applications where you are going to fight against just thermal noise in your detector, and for that we want to push that limit as well.
Okay, thank you.
Okay, now Nicoletta, please.
Thanks, Andrea, for this very interesting talk. I have a question about the role of the size of the pixels, because I can see the role of the orthogonal direction in the signal, but is there a minimum size that you can achieve? Is it better to have larger pixels? And how, if you have studied this kind of property?
Yeah. Let me take.
If you want the slide on,
it's a good question, and again, it's difficult to define the optimum because it will depend a lot on the system that you're using, on the problem that you have. So first of all, what are the limits? In fact, we're very far from any limit. So we could, of course, make them larger, but we could also make them much smaller. At the moment, these are about so this total size is 1.5 to 2 millimeter. So we're talking about 750 micron or so. And on this direction is 100 micron or so in the order of magnitude, maybe 100 to 200. We could make them smaller. Of course, you can make detectors that are 10 by 10 or smaller. There is no, at the moment, so the trade-off that you have is if you make it larger, typically you will be able to collect more light. And that will improve your signal to noise ratio. So that is good. However, of course, at some point your chips become larger, and then you can make many less on a chip, and that will eventually bring the cost up. So making them smaller, I don't see actually a reason to make them smaller right now, unless you want to increase the number of pixels in the array. And there could be cases where you would like that, if you can deal also with the electronics. And then you may be pushed more in the direction of course, smaller pixels. And of course, the limit is, you know, the CMOS cameras have pixel now in the micron, a few microns range. So that's, there's a lot of room to go in that sense.
So now there is a question from the YouTube chat. It's from Elizabeth. Thank you for interesting talk. I would like to know how your system overcomes sample metric issues for biosensor applications. I mean, in terms of clinical samples. Thank you.
So, sorry, sample metric issues?
Yeah, that's what she wrote. I know for biosensor application. I don't know, maybe if you do not understand the question, we'll ask Elizabeth to formulate a little bit better. I don't know.
Maybe, yeah, that would be good. I don't really get.
So maybe it's not so clear, Elizabeth, what is matrix issues, sample matrix issues.
Sample matrix. Okay, yeah. Okay. No, I understand that. Yeah, okay. Sorry. Sample. So basically, the specificity. So that is, of course, the issue in any practical biosensor, and there's no general solution for. Universal solution. And certainly, this is not what our area expertise. So my point here is we developed.
Andrea, I think you got it, but Elizabeth said, I mean samples like serum, blood.
Clear. Yeah. So the problem is, of course, we typically do this experiment first in a very simple matrix. But then eventually you have to go to blood or urine or similar, and then you will have lots of complication because there are, of course, many molecules, and then you have some spurious, basically unspecific binding, and on top of that you have big change in refractive index and other things. So this is true, and this is the main issue. So this will basically put this would make that only some biosensing problems or some biomarker recognition problems are actually really doable. But that is a common issue to all optical biosensors. So
essentially, Andrea, just for me, it's just selectivity. The point is selectivity.
Yeah, specificity, yes. How specific? Yeah, whether you can detect a certain molecule in a complex matrix where there would be hundreds, thousands of different molecules, and of course, some of them could still bind, or some of that could just deposit on the surface, even though you try to avoid that. And that has to do a lot with how you, what is your functionalization layer, how you treat the surface, how your fluidic system, and so on and so forth. So this is I don't have personally a solution to that. Of course, some problems will be doable, some others not. The key point for me is to provide with a optical readout system that can enable basically to a low-cost readout for many of these biosensors. I mean, we are doing this with photonic crystal. It could be done even with some plasmonic structures. And then, of course, other people or other colleagues. So we are also working on this at TUE, but then we need it's a multidisciplinary collaboration.
Yes. Now, Marco, please.
Yes, can you hear me?
Yes.
Okay, thank you, Andrea, very much. This really speaks to me, what you're showing. So my questions are concerned mostly the filter, okay? The detector, the InGaAs detector is the same. They're photodiodes, they're all identical. And then you have control of the filtering through the Fabry-Perot cavity thickness, basically. But in order to integrate this Fabry-Perot, you integrate this Fabry-Perot around the photodiode.
Yeah.
Why not? And you have to do this flip, chip flip, which is not easy.
Yeah.
And at top edge. So you have alternative solutions. For example, doing Bragg mirrors by epitaxy, but I guess it's very expensive. And then you need to have different. So why not gluing a Fabry-Perot cavity like the one you do on top of?
Yeah, it's definitely a very good question. We look at all this, and maybe this is, I don't know if this is the final thing, but I can share some considerations. So first of all, Bragg mirrors will not make it. And the reason, epitaxial Bragg mirror, so the index contrast in this material is very weak. So that means that your bandwidth of the Bragg mirror is very poor, it's very limited, and that absolutely is not enough for covering this broad range. And even if you have dielectrics, Bragg mirrors, it will also still not do it. So metal actually is very important here.
Yeah.
Now, then how to integrate the filters, and there are many different ways, and there are, particularly in the silicon, there are commercial arrays of detectors integrated with filters, basically, either gluing or fabricating separately. I don't know all the details. So partly it's possible, but gluing, we realized that gluing was not a good idea because it would be very hard to actually, gluing, say, not microscopically, but gluing really physically, assembling them, then you would have a problem how the coupling between the filter and the detector will not be perfect, so that there will be some spreading of light on the adjacent pixel and so on. This is actually quite tricky. And of course, you could fabricate a Fabry-Perot on top of a pixel, of a detector. The issue that we see is that because you need this metal, transmission of a Fabry-Perot, of course you have loss because of the metal, and you have to go through both metals. So we see that actually the absolute responsivity that you can get in that case is not as high as the one that we have with our structure. So that is the main reason why we went to these resonant cavity structures. But indeed, there are different ways of doing this. Bottom line is that you have to come up with a really technology solution. What is really, I think, really, really important is that you need a way to integrate this and fabricate this in an integrated way. And anything else, or putting filters, assembling filters on top of detectors, it has a limit in terms of both scaling of, say, the cost, but also the size of the array that you can make.
Thank you.
Okay, so my expert, be prepared. There is another round of questions. I make a comment in the meantime. So, with our eyes, we can recognize 1 million of colors, more or less. And they tested this with just notable differences. So they compare two colors. And when you say the one is different to the other, it means that and this looks like your spectral shift in the sensor. So you just look for notable difference, but you have 16. So I expect that the sensitivity increase exponentially with the number of of sensors. So you can, in principle, have extremely high sensitivity indeed. What do you think?
Yeah, indeed. So this is, I think that we could define this multidimensional color for the 16 pixels, and then we could ask the question how many colors we can actually detect in that sense, and that will scale, as you say. Of course, it also depends on the signal to noise ratio, and that is very dependent on the problem. And on the particular experimental situation. So it's difficult. I mean, for the eye, you can do it, well, in a given, of course, also for the eye, you have to actually fix illumination condition and so on. But that is, so it will not be one number. It will depend on how you use it. But yes, it's many colors indeed.
Okay. Now, Anna. Microphone.
Sorry. Andrea, you are an expert in indium gallium arsenide. And my question concerns the fact, if you want to realize some similar device in a different spectral range, so moving to the visible or the UV, do you see any problem in changing which kind of material are you thinking about? And do you see any further problem in the realization of the system?
Yeah, yes, we do. And I can share this, so because we have looked a bit into this. The main problem, if you keep the same detector structures or resonant cavity detector, you will be killed very quickly by the absorption of the material which is not supposed to absorb. So this is based on the fact that we have an absorber and we have some other layers which do not absorb. And now, for example, the contact layer. I mean, even in a photodiode, we need contact layers. And then the specific problem we have right now is that at wavelength below 800 nanometer or so, well, indium phosphide start to absorb a lot, and then you have a lot of loss. And that loss, first of all, is lost actually because you don't generate useful photocurrent with that, useful carriers, but also it degrades the cavity. So our bandwidth becomes very large, and then we don't resolve the peak anymore, and then it doesn't work. So, and that will apply also to silicon. Plus with silicon, I'm not sure you could do this trick either. You could think of going in the direction of gallium nitride. Then, of course, you can be transparent through the visible range and other materials, more fancy materials. But so, yeah, you can do this as long as you have a combination of an absorber and a transparent material in the wavelength, and a mirror, of course, in the wavelength of interest. Of course, for different wavelength range, you could also, well, use other approaches. And I mean, in the visible, I mean, just making this system in the visible, I want to mention there are companies which are offering multispectral arrays in the visible, and they are not monolithically integrated, I believe, but so they are quite big, let's say, with much larger dimensions, but probably they still do the job. So only the visible is not interesting for me because it already exists basically.
Okay.
I think extending that would be more interesting. So extending, I mean, combining near infrared and visible, maybe near infrared, mid infrared, that would be very interesting.
Okay, thank you.
Nicoletta, microphone.
Yes, this is just a curiosity. Probably you already said that during the talk and I missed it. In order to reconstruct the spectrum, you need to know the incident spectrum of the incoming light. Is it right?
To reconstruct the spectrum, I don't. To reconstruct the effects of the material, yes. Or I just need to keep a light source the same. So when I this is always based on a calibration. So when I train my my detector or my sensor, I need to make sure that the light source I use there will be the same light source as I use then with the unknown data.
Okay.
Not only the light source, but also the rest of the system has to be the same.
And just a curiosity, what kind of source do you use? And since you mentioned that it can be used in, for example, industrial situations and contexts, how can the operator reproduce the experiment? I mean, is it included in the package also the illumination source, or can it be changed accordingly?
Yeah, so both are possible. In the module at the moment that is commercialized by Multispectra, there is a light source included, and it's just a simple halogen lamp.
Okay.
And that is typically what is used in this near-infrared spectroscopy. You can, of course, just decide to use the chip. If you have a more complex system where you want different light source, more powerful or anything, you can just use the chip only. And then the light source is, well, maybe an issue at some point because we need this very broad spectrum. And at the moment, this is only possible with these halogen lamps. And they are, not ideal for many reasons. A bit large, not very stable, not extremely stable. So you could think of better light sources, but these are not. There is research on this and there are developments, but these are not, at the moment, not commercial available.
Okay, Marco?
Yeah, a very simple question. I don't know if you can answer, but if Mantis Spectra were producing 100,000 of this small spectrometer, how much would they cost? $1, $10, $100?
I will just take the easy way and tell you that I cannot answer, at least not as in representation of Multispectra. What I can say is that if you just look at some devices that we use daily, right? And okay, of course, silicon, okay, CMOS camera. Of course, it's extremely complex and still the price is very low. So then you tell me, well, this is a 3.5 device, so it's not silicon, it's more expensive. True. However, at the moment you can buy VCSELs. I mean, if you have an industrial application, a big market, you can buy vertical cavity lasers for less than €1. I think much less even. And these are actually even integrated in some smartphones, those that can take a 3D image. So that makes me think that in this business it's just market size. So if somebody comes with a really large market, then the price can go really down, but it's only possible if you have this extremely large volumes. That's also the downside. So I'm not claiming, this is a €1 sensor because, it depends on the volume.
Yeah.
But again, this is my opinion and not Multispectra's.
Yeah, but my question is probably joined to Marco's question. So I think in the separate waste collection, if you can recognize the kind of plastic a given bottle or a given box is made of, maybe it's very important. So I think in that direction, the market is huge. I don't know if you have competitors in this kind of business, but this looks to be very good, very important.
Yeah, of course, we have been, looking into this. I mean, one thing to realize is that spectrometers are actually used right now in sorting system, in industrial sorting system. When a plastic comes and then needs to separate it, they do this spectrally. So it's well known. The question is a bit, would you do it at home, right? Do you need it at home? Question number one. Because then it means you need to come with your 6 bags and go. And at the moment, at least in the Netherlands, we don't have the kind of plastic collection system. Second, of course, is that if you're talking about bottles, well, you can read it on the label what it is. So in that case, you don't need something like this. So yeah, it's probably in the future. I would agree with you that there is probably a very interesting opportunity, but right now I'm not so sure.
Okay, so I think we thank the speaker and all the experts and the audience. Thanks, Andrea, and I see you next week. Bye-bye.
Thank you very much. Bye-bye.
Bye, thank you.